Dear chairs, dear audience, my name is Réka Tóth, and I would like to talk about the work that I participated at the laboratory where I work. We investigate the changes of tissue pH in the ischemic red brain during spreading depolarizations. So I would like to present the methods that we use to carry out our experiments. The brain is a really important organ for the human body as the center of the nervous system. It's only 2% of the body's whole weight, but it takes up 20% of the body's oxygen use. Another proof of its importance is the incredible capillary system of the brain, since the overall length of it is 600 kilometers, and it provides 20 square meters of surface of endothel for constant nutrient exchange. The constant perfusion for the brain is crucially important because the brain has no metabolic reserves. It cannot store glucose or oxygen on its own. An interruption of blood supply for the brain just for 10 seconds results in loss of consciousness, and if the interruption lasts for 2 to 10 minutes, it will lead to brain death. This represents really well the importance of this topic. This is why it is really important to research the diseases connected to it. One of the diseases is stroke, and it can be seen in these pictures what damage it can cause to the brain. Stroke in the last few years stepped forward from the third to the second place among the most frequent causes of death. Each year, 15 million new strokes are diagnosed, and Hungary's part of it is 40,000 new cases every year. Besides that, in most of the cases, the patients who suffer a stroke event later, to some extent, will become disabled. The age group at highest risk of having a stroke are the elderly people. 75% of patients is more than 65 years old, and this is due to the fact that the probability of stroke increases with age. In the case of ischemic stroke, the primary injury is caused by the obstruction of a cerebral artery. The center of the injury will be the core area where the neurons are dead, and around this region there will be the penumbra, where the neurons might still be viable after the blood supply is restored. Unfortunately, we have very limited possibilities to fend off the primary injuries because the therapeutic window is really short, so the success rate is very low. Hours, days, or weeks after the primary injury, secondary injuries may arise, which are more important from the therapeutic point of view because we have time for intervention, and with this we could prevent the further damage, so the stroke outcome can be efficiently improved. Cortical spreading depolarization, to which I will later refer to as SD, is such kind of secondary injury. SD is a wave of large negative shift of the slow electrical potential of the brain. It propagates slowly across the gray matter with a speed of 2 to 6 millimeters per minute, and it spreads from the core towards the penumbra region. After stroke, it usually occurs spontaneously. Its characteristics are decomposition of ion homeostasis, metabolic changes, and it's also accompanied by characteristic hemodynamic responses. There is also a transient acidosis with the SDs in the tissue. While in the intact physiological brain, these SDs can only be elicited experimentally, and they do no harm to the tissue. The SDs that occur spontaneously after stroke may increase the extent of the injury under ischemic conditions. The importance of pH regulation in the brain is that it modulates the sensitivity of certain channels, enzymes, and receptors. Also, the acidosis reduces the excitability of neurons, which might lead to cellular dysfunction. We know that ischemia itself causes transient acidosis of the tissue due to the piling up of lactate and carbon dioxide. We also know that SD is associated with transient acidosis. So we came up with a hypothesis that SD-related acidosis is superimposed on ischemia-induced acidosis, and together, the more pronounced acidosis leads to a worse tissue damage. To prove our theory, we set it as an aim to determine the SD-related pH changes in the ischemic cerebral cortex. And since we know that the age group at highest risk are the elderly people, our other aim was to investigate the impacts of aging on the dynamics of pH changes. In our group of research, we have a really unique imaging method, which we use for imaging the intracellular pH changes of the tissue and also the changes in cerebral blood flow. It's based on the cooperated operation of 2 CCD cameras attached to a stereo microscope. We have one LED emitting green light, another LED emitting red light, and also a laser diode. One of the CCD cameras is completed with an emission filter, and it allows only waves between 600 and 640 nanometers to pass through. This is, which is responsible for recording the light of the fluorescent dye that we use. So the imaging is based on a pH-sensitive fluorescent dye called neutral red. We administer it to the rats intraperitoneally, and during the 40 minutes of incubation time, this dye will bind to the inner side of the cell membranes of the neurons, and then we will excite it with a green-red LED light, and it will emit red fluorescent light. The cerebral blood flow is measured with a laser spectral contrast analysis. In our experiments, we performed them on young adult male spray dolly rats under anesthesia of isoflurane. We monitored the blood pressure and blood gas with a cannula in the left femoral artery, and we also prepare a closed cranial window on their parietal bones for the imaging. After 10 minutes of recording the baseline, we elicit 3 SDs with the topical application of potassium chloride in 15-minute intervals. Then we create transient ischemia with the occlusion of the common carotid arteries on both sides. During the ischemia, we elicit 3 more SDs. Then we cancel the occlusion and start the reperfusion with 3 more SDs. These representative videos were made to show the changes in the tissue. Here, the field of view can be seen. The second figure will show the changes in the cerebral blood flow, while the third picture, I hope it will be visible, but it shows the changes in the intracellular pH with the fluorescent dye. So the fluorescence will increase as the pH of the intracellular space will move towards acidic. So lower intensity means alkaline, while higher intensity means acidic. These videos are synchronized. So first the fluorescence change will be visible. Then with a short delay, the appearance of warmer colors here will show the increase in the blood flow. Okay. So in another set of experiments, we created 2 open cranial windows on the rats. In the caudal one, we elicited the SDs, while in the rostral one, we implanted a DC electrode, a pH-sensitive electrode, and a laser Doppler flowmetry for recording the signals. We had 2 experimental groups, a young and an old one. So now I would like to show some data that we've acquired from this setup. Here, a full recording of an experiment can be seen. The upper curve shows the changes in the cerebral blood flow, while this one under, it shows the changes of the extracellular pH. Later, we analyzed the SDs during the baseline, during ischemia, and also during the reperfusion. It can be nicely seen that the acidosis caused by the SD is accompanied and followed by a functional hyperemia. Our results show that the degree of SD-related tissue acidosis during ischemia was significantly bigger compared to the baseline. However, we can also see that in the old experimental group, this degree of acidosis was significantly smaller compared to the younger rats. About the magnitude of SD-associated hyperemia, we can say that in all three periods, so baseline, ischemia, and reperfusion, the old rats showed a lot smaller hyperemic response compared to the young ones. And in the old group, between the three periods, there was no significant difference in the magnitude of hyperemia, while in the young ones, during ischemia and reperfusion, the magnitude was significantly smaller compared to the baseline. So, to sum up our results, we can say that ischemia increased the extent of acidosis in both groups, but the changes of the pH were significantly smaller in the old group. Regarding the cerebral blood flow, we can say that it dropped significantly with the induction of the ischemia, but it also lasted longer. However, the hyperemia was found to be of smaller amplitude and shorter duration in the old group of animals compared to the young ones. So, in contrast with our expectations, we found that the more severe ischemic damage in the old brain is not due to the deeper acidosis, but to the significantly smaller hemodynamic response associated with the SDs in the old rats. I would like to thank my mentor, Eszter Farkas, for the constant support, and all my supervisors, and thank you for the attention.